9 out of 10 children, if you label them falsely as possibly autistic, as high risk of autism, there is a risk that this will have an influence on the behavior of the family towards these children and actually increase the risk that they will have some kind of psychological problem, just because they are regarded as potentially autistic. I'm sure you remember those experiments where children chosen at random in a class were indicated the teacher as especially bright or as especially dull, and indeed the ones which were indicated as especially bright got better results, because actually the behavior of the teacher towards those children was different, even though there was no objective basis for that. So it doesn't actually look like a good test. And in fact, there was a lot of reaction to this announcement. I myself published a paper about it, and there were strong worded comments by a number of autism charities. But maybe one of the most strongly worded criticism was published in a magazine which actually is published by Handicap International, which is a well-known charity. They have managed to more or less outlaw landmines, for example. And so the title, well, this article, the title of this article was Who Profits by the Crime? And you see here a dollar bill with pills. So the idea is this is, well, and the article itself, if you can read French, it's really very strongly worded, and it says that this company is completely responsible, that they are just looking for money, that they don't worry about what's going to happen to the false positives, and so on and so forth. And actually, the company had a libel suit against this journal. It's shown here. So December 2006, and basically it asked for 100,000 euro damages, which is quite a lot of money, especially for a small publication. It can be enough to make it problematic to survive. I was actually asked as a witness to the trial. Unfortunately, I couldn't be a witness because I was in Taiwan. But not as a result of my absence. The company won. The company won the libel suit. They didn't get 100,000 euros. They got what we call one symbolic euro. But they did win in principle, and they did get publication of the judgment in 2 or 3 magazines. So even though they didn't get a big pot of money, they actually won something. So even if the paper was indeed a little inflammatory, it could have been worded more carefully. It looks like really a paper that someone wrote and then just published without sleeping over it and having another look. But still, it's really a problem if when a newspaper criticizes a company, this company can actually win a libel suit against the newspaper. It reminds us of what happened with tobacco. The tobacco companies really managed for 1 or 2 decades to prevent the link between smoking and lung cancer from being acknowledged. And of course, during this time, they made a lot of money, and during this time, a lot of people got lung cancer. And so this is an example of the kind of advertising that was common in the 50s, involving doctors and reassuring people that smoking was okay, since doctors smoke. but there was a lot more action, like bribing officials, publishing false scientific studies, persecuting scientists who published studies showing link. I mean, it was really very dirty business. I'm not saying that Integragen is as bad as Philip Morris or whatever, but there is some connection. Okay, so actually the test did not come out in 2006, and did not come out in 2007, did not come out in 2008. And in the meantime, there was more research on the genetic correlations of autism with better and better technology. Because now we can do whole genome association studies where you look at 10,000, 100,000, even now 1 million markers all across the genome, and look whether there is any association between certain alleles of some of these markers with the condition, looking at a few thousand patients, a few thousand controls. And one of the earliest and largest studies was published in 2007. So involved 7,000 people, 138 authors, looking at 10,000 markers all over the genome for all these 7,000 people. And this is the kind of results they obtained. So what it shows is a scan across the whole genome, the various chromosomes from 1 to 22X. And here, well, you can't quite see the scale, but it's the probability that the result is not false positive. Because of course in these studies you are looking at many patients, many markers, and there is always any association you see could be due to chance. So you have to apply very strict statistical criteria to be reasonably sure that what you see is a real association and not just a random fluctuation. So this is the statistical probability that the result is solid, and this is the line above which the result is considered pretty sure. Of course, never 100%, but with a really good chance of being statistically significant. So you can see there are lots of signals on lots of chromosomes, but really only one that is above the limit. So indeed, this genetic influence on autism must involve many genes, and to find them it will require different experiments or even more people, more SNPs, and more work. This is already a very big piece of work costing millions of dollars. Also, during these periods, we realized that single nucleotide polymorphism, so-called SNPs, are not the only type of variation in the human genome, that there are also differences involving small, or in some cases fairly large, deletions or duplications, pieces of DNA that are missing in some people, that are in several copies in other people, so-called CNV, copy number variations. And indeed, some of these CNVs appear to be linked to autism. They are a minority, but there is, in some cases, fairly significant linkage. So it's not only point variations, but also some deletions and duplications that seem to be involved in autism. And then also, it was found that in some cases, particularly in some family studies, you have de novo events, alterations or variations that are not present in the father or in the mother's DNA, but are present in the child's DNA. And so that must be a result of a new mutation, a new point mutation, or a new copy number variation that happen with the child. In some cases, families where you have two affected children, you find the same de novo change in both children. And so in that case, it's pretty strong evidence that this has something to do with the syndrome. So we have made progress, but the picture has become more and more complicated, not only point mutations, but also deletions, duplications, and not only inherited changes, but also de novo changes. So we still have this relative paradox. We have a syndrome that is largely determined by genetics, but we have a lot of trouble finding the genes, even with the most powerful technology. And of course, this means that developing a really useful genetic test is going to be very difficult. Now, this problem here, this elusive gene problem, is not limited to autism, and it's actually quite general. And it's the so-called missing heritability problem. We are indeed now in a situation where we have a number of diseases or conditions where there is clearly a very strong genetic component, where using new technology, one has been able to find a number of genes that have some influence on the disease, but we don't account for all of the heritability. For example, if you take height at adult age is a very strongly inherited component. The heritability is something like 0.8, 80%. Of course, heritability of 1 means it's entirely determined by genes, and like Huntington's chorea, for example, and the heritability of 0 means it's entirely determined by the environment, like through infection. So for height, the heritability is 80%, very strong, and one has by now found, I think, more than 50 genes that are involved in heritability. But these genes, all these genes taken together, they only account for less than 20% of the heritability. So we are missing a lot of it, and well, there are technical reasons for that. I'm not going to go into detail. It's one of the big problems in human genetics right now, finding where is the missing heritability, and it has to do with the frequency of variations in the genome and the limitations of the methods we are using, and also probably some things we don't understand yet. But this is again, this is quite a general problem. Also with purely organic diseases like Crohn's disease or even diabetes. So it's not limited to more psychiatric diseases. Of course, then you have the problem of, especially with something like autism, it's probably very heterogeneous. What we call autism is probably a number of different conditions that we lump together. And so, of course, when we take patients suffering from any type of autism, it makes it even more difficult to find the genes involved. And then we have this involvement of de novo mutations, which makes things more difficult. I think, for example, for Crohn's disease, this is beginning to be worked out. I mean, this type of approach was sort of plateauing at 20 or 30% of the heritability, and now newer studies that look at rare variations are picking up. Okay, so this is the landscape. So what has Integragen been doing in the meantime? Actually, even though I was somewhat hostile to what the company was doing, I have had frequent interactions with them. I visited them a couple of times. And they have continued working on autism, and in fact, in 2005 it was one of their topics. By now it is their major topic. So, this is actually an interesting question. Why would a company devote most of its activity to such a difficult question where it's unlikely that they can make a good test? And we'll come back to that. But they have published a respectable number of papers and poster sessions, oral presentations, and so on. There's a very abundant literature about the genetic aspects of autism, so they are a small part of that, but they are contributing. And fairly recently, they had a paper which describes what you can do with 4 genes to determine the autism risk. So I think this is a paper. So this is their most recent paper, published in 2010 in a new journal, but it's supposed to be a peer-reviewed journal. And essentially what they do, like in this article, is to describe in detail the test that they announced in 2005. In 2005, they announced a test based on 4 genes. Only one of these genes was published, and 3 others they just referred to in their advertising, but they didn't say what the genes were, what the results were. So actually, in this paper, they do describe the 4 genes in detail, and more importantly, they show really the results of the test, and that's what we're going to look at. So this is we'll spend a little time on this graph. So what they did is take a number, I don't remember the number, but a fairly high number of children who had already been diagnosed as autistic, and an equivalent number of controls. I think it was like something like 200 children in each category. And then they performed the test on all of these children, autistic and controls. And here they plot the results according to their genetic score. What they call their genetic score is the result of the tests, and 8 is the highest score giving the highest risk, and of course 2 is the low score giving the lowest risk. And so what you see here, the black bars are the autistic, the children that have been diagnosed as autistic. Of course the test was performed without knowing which child was normal, which child was autistic. And the gray bars are the normals. And it's expressed as the percentage. So for example here, this means that for a genetic score of 5, you find 31% of the autistic cases and 38% of the normal, of the control individuals. So what you can see is that as the score increases, you have proportionally more and more autistic children. So for 8, you have almost only autistic children. For 2 or 3, you have mostly normal children. But it's obvious that the separation is not very good, not good at all, because what you would hope for is that above a certain score, almost all children would be autistic, and that's not at all the case. So if you take this data and you compute specificity, sensitivity and specificity, which is a standard way of assessing the quality of the test. Let me just have a little look at that. So this is, I'm sorry, this is in French, but I'm sure you can. So this is, here the test is negative, here the test is positive. These are affected individuals, these are control individual. So ideally, all the affected will test positive, none will test negative, and also ideally all the controls will test negative and none of these will test positive. Of course, in reality, things are never perfect. You have a small fraction of patients who test negative, so these are the false positives, and you have a small fraction of controls who test positive. These are the false positives. So sensitivity, what we call sensitivity, is the fraction of affected who test positive. Ideally, it should be 100%. For example, for a commonly used HIV test, it's 99.4%. And specificity is a fraction of controls who test negative. Again, ideally, it should be 100%. For an HIV test, for example, it's 97.6. When you are doing a screening test, like HIV test, you'd like to miss as few positives as possible. So you want the test with very high sensitivity. If the specificity is not very good, that means you will have some people who will test positive when they are in fact negative. Of course, when somebody tests positive with the screening test, then you do a second test, which is more expensive, more slow, and which will eliminate the false positives. So people will just be anxious for a week or two, and then they will get a good result. But at least you're not going to miss people who are actually infected. So it's always a balance. If you want very high sensitivity, you may have to compromise on specificity and vice versa. So in this case, really, and this things looks very bad, because if you take as positives only the children with a score above 6, so with these, just these, you're going to have only a small fraction of the affected children, something like 17%. And then your specificity will be relatively good. You won't have too many false positives. But if you want to increase the sensitivity, if you take all scores above 6 as positive, then your sensitivity is better. But now your specificity has gone down a lot, and you're going to classify many controls as positive. So whatever way you do, the test is not really good. However, in fact, this test or a closely similar version has been marketed. It's been marketed late last year by a company called, the name escapes me, I'm sorry. In US, Transgenomic, which if you're familiar with the field, it's a company that's known mainly for making the so-called WAVE system for S&P scoring, a system that's largely out of date by now, and the company is probably trying to reinvent itself as a genetic diagnosis company. But anyway, so they marketed this test, which they called the A-risk, familial autism panel. It was actually based on 8 genes, so the 4 preceding plus 4 new genes. They had a so-called white paper, which is actually a paper that has not been published in a journal, that's written as if it was a real paper, which showed slightly better performance. So this was indeed announced and marketed according to the site of IntegraGen, but I was surprised to see recently that it has now disappeared. The addresses that recently had just now lead to a general description by IntegraGen, and so apparently they tried to market this test and probably either did not get any orders or were criticized. I don't know the inside story, but the end result is that the test is not on the market. Okay, so this is, I think I'm coming to the end. So what about the future? As as this test, well, a good genetic test for autism risk would certainly be useful again, because if it allowed you to detect autistic children at an early age, it would help to have better treatment and to improve the quality of life for these children and their families. However, at this time, as we've seen, the current tests are very limited. They don't really discriminate children at risk and normal children with any efficiency, and there is a serious risk of misuse of these tests. Well, maybe in the future, since autism is probably a mixture of different syndromes with different etiologies and maybe different genes involved, so maybe in time identification of genes that have an influence will allow the understanding of a number of pathways, and maybe allow to subdivide autism into several different syndromes, which may have different therapies, in particular in terms of drug therapies. So this may help not only detection but also therapy in the future, but we're not there yet. And so, question one might ask is, given this fairly hopeless current situation, why would a company put all its efforts into this type of work? Well, this is a case where actually the market is waiting for our products, and the market is quite significant. There's something like 30,000 new cases of autism per year just in the US, about as many in Europe. It's just 1% of live births. And if such a test is used, it will probably be used not only on those who turn out to be autistic, but on a number of likely cases. And a fairly complex test like this is usually sold around $1,000. So it adds up to quite a significant market. And in fact, both the parents and many doctors are really waiting for a test. They would like to have a test. They hope, they expect, and they are wrong in this expectation, but it's quite ingrained, that the test will really, because it's a genetic test, it's going to be black and white and tell me this child will be autistic or this child is normal. This will never be the case, but this is a widely spread misconception. And of course in the US, doctors are always afraid that eventually they will be accused of not having done a test that could have had possible results, so in the doubt they will order the test. This is already happening for breast cancer prognosis, for example, so it's certainly a factor. So, conclusion, even though we've made really a lot of progress in analyzing genomes, and we can now sequence genomes, and we can do a lot of things, and we have solid scientific data, because those associations that are found, they are valid and reproducible. They just don't explain enough. So we still have a lot of problems going from the genes to the phenotype. I really like this drawing where the MDs are looking at the gene sequence. It doesn't really help them to understand why this patient is feeling so bad. And I think I have another take-home lesson. So this is another drawing which I think is really very, very good. And so I think this shows the contradiction that may exist that exists in some cases between business interest and a responsible approach to diagnostics, and which means that we really need effective regulation, which is not easy. And I think that